Okay. Hello, everyone. I will try to show you in this second part of this webinar how can you employ this integrated methodology to analyze a special nanostructure. This talk has two sections. The first one is how to apply this methodology in real cases. Actually, the first one is about the quantitative high-resolution STEM analysis of low-dimensional materials. And the second methodology is about to analyze structural defects with aberration-corrected high-resolution TEM. The second section of this part contains the quantitative high-resolution STEM analysis in practice, and I will show you in brief our platform that we developed here to analyze high-resolution STEM imaging. Professor Dimitrakopoulos gave us the general methodology we have here to analyze nanostructures, and now I will try to show you the parts of this methodology we employed to analyze to perform compositional and strain analysis of ultra-thin InGaN quantum wells. And the second case study is the structural mobility of Shockley partial dislocation in gallium nitride. Okay, let's move to the first one. Let's give you some brief introduction about these ultra-thin quantum wells. These materials, these novel materials, can be used for bandgap engineering, covering all the visual spectrum, and they are promising for spintronic and for spintronic application quantum computing due to their potential topological insulator behavior. But in order to use these materials, we have to grow such materials, and it's extremely difficult. Here I will show you. You can see now that what is such wells. We have here a high resolution conventional TEM image that show uniform and continuous quantum wells. These wells were grown by plasma-assisted molecular beam epitaxy, and they have periodicity 5 or 3. In order to analyze them, we used a probe-corrected high-resolution STEM microscope, and the quantification of high-resolution STEM was performed using a combination of multi-slice image simulation, peak finding, and GPA. Of course, the cornerstone of this analysis was the atomistic calculations that gave us the structural and chemical models. These models were created by using empirical potential calculations and density functional theory. And now you can see what are these quantum wells and what is their thickness. You can see here that we have quantum wells with thickness they are extremely thin. Their thickness is limited to 1 or 2 monolayers. When the growth temperature is low enough, you can see that the quantum well have thickness about 2 monolayers, while when the temperature is higher than the limit number, then this monolayer have thickness 1 monolayer. And you can see here a high resolution STEM image where the 1 monolayer quantum well is given. How we can measure the strain and the strain in such monolayers? The first method is the most known method, is a GPA method. We can extract strain information about this nanostructure by using GPA and using the proper mask size of the image. GPA is a well-known method. It works in the reciprocal space, in Fourier space, and the critical point in this method is to have a mask size able to analyze the strain you want. But here we have to mention that the mask size is the parameter that defines the spatial resolution of this method. In this case, we used a mask size with radius equal to G0002 reflection, and we have a spatial resolution equal to cell lattice. So this method is limited to extremely thin nanostructure. Therefore, we have to implement the peak finding method. Professor Dimitrakopoulos gave us the initial stages of peak finding, what is peak finding and how can employ. And now I will show you the steps of this methodology. First of all, we have images like this, and we want to assign the center of peak, the center of these bright spots. First of all, the initial positions of the bright spots are determined directly by its very common way. Then we have a first round of refinement using the intensity weighted center of mass, and we have a second round of refinement by fitting 2D Gaussians. In this case, the center of these spots are defined by the center of the Gaussians. So, if we have the positions of the peaks, then we are able to perform strain analysis. But it is important to perform peak classification in sublattices. In our case, we have an hexagonal lattice, and hence we have to choose the proper peaks in order to group them in sublattices. We have here the A2 vector, that is this value is equal to cell lattice, and we determine the lattice strain using this math type. Of course, the material system dictates the crystallographic constraints, and in each case, we have to take into account the crystallography of the system. And now let's see how can perform peak finding in an experimental high-resolution STEM image. First of all, we have to mention here that peak finding is performed and the peak positions are determined, and then we calculate the lattice strain by the former math type I showed you in the previous slide. Then, in each peak position, we assign the set distance from the previous one, and following this procedure, we can find how the cell lattice parameter is changing in this image. As you can see, the strain is confined in one monolayer. I have to mention here that each rectangular box has its center a peak position from the image, and in this way we can determine the average strain in this monolayer. We have here 3.5%. By employing this technique in multiple areas, we can find the average strain of the samples. You can see here cases for 1 and 2 monolayers, and I want to keep it in mind and how we can translate now these lattice strain values to composition. In order to perform this analysis, to translate the strain to composition, it is important to perform empirical potential calculation, perform energetic calculations in order to have, firstly, some structures in order to find the strain behavior of such ultra-thin quantum wells. Density functional theory is an ab initio-based method from first principles and used in order to check the empirical potential, to check if our modified embedded atom method is accurate enough to describe such structures. And the next step is to model structures that that cover all the cases you can find. So in this case, we developed models with one monolayer thick quantum wells, and the thickness and the composition in this monolayer covers spanning the whole compositional range from 0 to 11 to fully covered with indium nitride. In order to be accurate in our analysis, we have 2,000 supercells overall, and we have different random distribution of atoms in such structures. Of course, the dimensions of such structures must be large enough to be close to realistic dimensions. Of course, possible ordered distribution must be considered, and therefore such cases were also considered. So now we have all the possible cases, all the possible models to analyze them in order to find the strain behavior. So after the energetic calculations, we analyze the data, and we determine the strain behavior, the strain along z-axis for one monolayer and two monolayer. These curves can be used as can be used in order to translate now the strain to indium composition. So this is the proper way to perform such analysis in the cases of ultra-thin quantum wells. Having now these curves, we can translate the previous strain data, the experimental strain values, to compositional values, but by considering only the thickness of the quantum well in each case. And now I will show you how you can estimate the composition, determine the composition by using not the strain values, but the intensity directly. The first part of this analysis, you have to find the imaging conditions of the experimental images. So you have to perform image simulations and find the proper imaging conditions, and then again with image simulations, we can determine the intensity behavior of the pure gallium nitride by changing the thickness. We can have now the capability to to find the thickness by simply know the intensity of the purely GaN regions. Another part to find the composition is to extrapolate the thickness in regions with composition. As Professor Dmitrakopoulos mentioned before, high angular dark field STEM images contain information about both thickness and composition. So it is important to subtract the thickness influence in regions with known composition, with unknown composition, and this procedure can be made by extrapolate the thickness to this region, as you can see in this graph. And now you can see how we treat all the images. For each image, the first step is to define the peak position, and then we assign to each position, to each peak position, we assign a Voronoi cell around it. The Voronoi cell, the intensity of the Voronoi cell actually contains the information about thickness and composition. And then we establish a comparison criterion, the intensity ratio. By having image simulations using structural models with known composition, we can determine now how this intensity ratio is changed by changing the thickness. And we have no Vega dependence. Actually, we create maps like them that can give us the intensity ratio values and by changing the thickness and indium content. By having a map like this, you can use it to analyze the intensity of the intensity ratio that experimentally observed. And so we can now to associate experimental intensity ratio to the indium content. And how is it possible? Let's see how you can do this work. First of all, we find in the first step the thickness of our region is the blue line, and then for this blue line we can find the behavior, the expected behavior of such wells by changing the indium content. By comparing now the experimental intensity ratio to this behavior, you can find directly the indium content from the intensity. Another part you have to take into account in this analysis is the cross-scattering effect. Actually, as you can see in the images, in the previous images, we assign a Voronoi cell in each atomic column position. Actually, we don't have only the peak, but a region with many pixels and with area. These areas are affected by the neighbor atomic columns, and so we have a complicated behavior. It is important to perform this analysis to find what is the artificial composition of atomic columns close to the ones with composition. As we know, in the case of one monolayer, for example, here, we have only one monolayer here with composition 33%. But as you can see, the simulations gave us that the monolayers close to the central one on the left and on the right, they don't have composition 0, but they have some composition close to 10%. And this means that we have an artificial composition of the adjacent monolayers to the central one. This must be considered in order to determine possible diffusion phenomena or possible enrichment of the monolayers close to the central line. In this color map, you can see how this the affected area is how it is changed by changing the thickness. When the thickness is higher, then this affected area is larger. And by considering quantum wells thicker than one monolayer, and therefore you can see that this affected area is much larger. So image simulation are important to determine this effect. And actually, this is the only way to determine artificial composition of the adjacent monolayers to the central one. Another part we have to take into account is the effect of the amorphous layer. In each STEM sample, we have the foil surface and a part of amorphization. We have amorphous layers in the surface. These amorphous areas are created due to ion milling, and we have to take into account the thickness of these layers in order to determine the crystalline part, and after that to determine the composition. So in this case, we modeled amorphous gallium nitride, and with multi-slice simulation with frozen phone approximation, we create images, and the intensity we measured, we subtract. Then this intensity was subtracted from the experimental images. And now let's see how we can have both strain analysis and intensity analysis. This is the previous case. We found from peak finding that the strain was 3.5%. The indium composition is determined close to 39% only by using the strain behavior, and by using the intensity now, we determine that the composition of the central monolayer is 40%. So these methods, strain analysis and intensity analysis, are converged. And the final part in this work is by comparing simulated and experimental composition profiles, it is possible to say if the adjacent monolayers have some artificial enrichment or they are a real enrichment. In this case, we found that we have only one monolayer quantum wells, despite the fact we have some composition, some enrichment in monolayers close to the central one. And now we move to the second part, the second case study that referred to the structural mobility of Shockley pass and dislocation in gallium nitride. This work is referred to high resolution TEM imaging and not in high resolution STEM imaging. In this case, we have actually a brief introduction about this work. We tried to see the behavior of dislocations under carrier injection. In situ beam irradiation is a possible way to see this this phenomena. In silicon carbide, for example, we can see that we have an expansion of the static fault width, something which leads to device degradation, while in the gallium nitride we observe that we have a reduction of the static fault width, which is a possible positive impact of for the devices. Professor Drakopoulos gave us previously all these mathematical expressions of optimized imaging conditions. As mentioned, we need to check some of them in order, and all of them, in order to have the best conditions for our work. we checked them all, and we found that the last one is the proper imaging conditions to analyze our dislocation course. First of all, when someone wants to test some of these conditions, we have to perform some analysis like that. We have to perform image simulations and see which condition provides better signal to noise ratio. You can see here that the image conditions we choose have better signal to noise ratio than the full aberration corrected condition. Then we have to test these conditions to see how behave our material under these conditions. These images are from image simulations, and you can see here that both gallium and nitrogen atoms can be dissolved, can be distinguished. You can see both red and green balls. The red balls indicate gallium atoms, and nitrogen atoms are indicated by the green one. But as you can see here, we have this. We see the light images, the nitrogen, only for thicknesses up to thickness to close to 7 nanometer, 5 nanometer. Yes, and we have here the intensity between the gallium and nitrogen column position. This behavior can be used in order to find proper conditions to see the course. So, in this work, the defects were studied in plasma molecular beam epitaxy grown InGaN heterostructure. The electron microscope observations were performed in Poland in the lab with Julita Smartkos-Groska using an aberration-corrected high-resolution TEM microscope. The HRTEM quantification was performed by using the multi-slice image simulation and GPA. And in this case, again, the atomistic calculations were important to correlate the experimental images with structure. So let's have now. Let's see now the possible structural models of such defects. You can see we have 5 models, and density functional theory calculations were used to provide us the information about which of these cores are the energetically favorable cores. The Glide gallium double period and Glide nitrogen single period are the most favorable cores, and we expect to find these cores in our analysis. But of course, we have to determine experimentally that. So we perform now a next round of a second round of image simulation, using now as input the structural models of our course. You can see here that the chosen conditions are able to distinguish the cores. You can see here an open core, and so these conditions, the chosen conditions, are the appropriate conditions to see the course of such dislocations. And here we have an image, an experimental image obtained in Poland that we have, and that you can see exactly the positions both of gallium atoms and nitrogen atoms, and this gives us the opportunity to directly assign its peak to the atomic column. So the core structure is directly reflected in the center of this image. So by using aberration corrected HRTEM imaging, under optimized imaging conditions, it is possible to directly see what is the core structure in the center here, and this is how important is to have the peak position here, because we have the atomic column. And now let's see a dynamical effect. This is the effect we observed. Julita Zmalkos-Gross observed this phenomenon along with Joanna Moneta. They saw that this stacking fault is shrinking in time, as you can see here, and we tried to see what is the behavior, what is the reason of this unexpected behavior. We have here a stacking fault, here we have a small stacking fault, and then the stacking fault is vanished. The first part is to analyze, using Burger circuit, the type of the dislocations. And the next step is to see what is the core structure by comparing now experimental cores, experimental images. This and this image are from experimental images, and the other one are from simulations under similar thickness. You can see that the upper one is close, is almost resembles to the glide nitrogen single period as expected, and the right hand partial is similar to the glide gallium double period. So we can find, by comparing simulated images and experimental images, the type of the core. And now I will show you which is how you can determine the mobile partial core. Actually, the images with different obtained at different time were aligned, and then we used the dislocation density tensor in order to locate the positions of the cores. We have to mention that dislocation density tensor is a tool that developed here, and you can, if you want further information, you can find them in this work of Professor Kersevlu. In simple terms, dislocation density tensor is the first derivative of the strain and has non-zero value only at the core region. So we can have a direct visualization of the core locations, and therefore, and hence we can see that the more mobile core is the glide nitrogen single period. So we can now have a model how two dislocations, the glide nitrogen single period and glide gallium double period, are recombined to the initial screw dislocation. All this works, as you can see since now, can be performed only by using computational tools. It is important to develop computational tools to analyze such images, to perform calculations, to obtain the behavior of the material, and many other things about that. But the most important part is the computational tools. In our lab, we developed the Artemis platform. Is a software ecosystem with multiple toolkits and an easy-to-use graphical user interface for quantitative, atomic resolution, high-resolution STEM analysis. Actually, this platform was developed by three atoms, by me, by Thanasis, and by Professor Dimitrakopoulos. We employed this in my PhD thesis, in the bachelor thesis of Thanasis, and we tested in many materials. And the short description of this platform is an autonomous platform, and it has an easy-to-use graphical user interface. It has a simple workflow to perform the analysis. It gives you the opportunity to have parallelization of the processes, and we have clever algorithms along with various methodologies. Another plus of this work is that we have both automatic and manual analysis. We have tools that you can have regions with different shapes, and the most important is that is an easy upgradable application. It is very easy to upload other tools and work all together. This platform can give information about nanoparticles, can give quantitative information, strain information, composition information about nanoparticles, quantum wells, core cell nanowires, and crystalline cluster. For example, nanoparticles embedded in a crystalline matrix with different lattices. And if you want, in the next minute, I will show you how to perform a quantitative atomic resolution STEM analysis using HRTEM. Actually, you have to follow the top-down workflow in each menu, and I will show you how to import the images. It's a the practical part of this webinar. I will show you how to import the images, how to analyze the strain, and how to analyze the intensity. Of course, I have to thank you all for your attention, and I have to acknowledge the project Nanoscopy-Gr, the project Ultimate. We have to say that the State Scholars Foundation support me in my PhD thesis, and further support by the project Innovation EL. And of course, we have to mention the important role of Aristotle University of Thessaloniki High Performance Computing Infrastructure. Thank you for your attention, and we can, if you have some questions, you can say now. If not, we can move to the practice part and the practical part, and I will show you how to analyze high-resolution STEM images.
Okay, thank you, Isaac, for this very detailed talk. And so now we can have any questions that you may have about this approach. We will show you, of course, now how this is done, at least to some extent, but what we are going to do in the future is that we are going to have a more detailed workshop, a one-day workshop on quantitative analysis of high-resolution electron microscopy images. Hopefully, in addition to us, we will have participation of other groups also who are doing computational microscopy. And of course, we are also going to do a practical workshop, either online or here in our lab, where we will show practically to students who are interested, practically how this analysis is done.